So we're going to talk about T cells. So T cells, again, remember, are more like the army. They're sort of this cell-to-cell kind of destruction. So if you look at this, here's a T cell. And inside here, this cell is infected. So the T cell is going to actually come up to it, come into contact with it. You can see it, it's contact, and then it's going to get destruction. But T cells have to be activated like others. Remember, T cells are going to attack infected body cells. And the reason we have these T cells is because antibodies can't get in there. So antibodies cannot enter cells. They only can attack antigens that are extracellular. So if you have antigens inside a cell like this, they're protected from the antibodies. The antibodies are out here and they can't get in. And as long as they stay inside that cell, they're protected. Well, it's kind of like having a bunch of bad guys in a house or a building, and you can't get into the building, but you want to kill all of these bad guys. Well, the easiest way to do that is to blow the thing up. You can just firebomb it or blow it up, and when you destroy the house, you also destroy all of the antigens. And even if you don't destroy them, the antigens are going to be released, and then antibodies can destroy them. And so that's the strategy that T cells use. They don't try to get inside the cell. What they do is they destroy the entire cell. So let's look at T cells. So remember, this is cell-mediated immunity. The other one was humoral immunity. Well, we only had one kind of B cell. They're all unique. They're all different from each other because they all have a different surface receptor. But there's only one kind. There's only one type. That's not true of T cells. When you look at T cells, there are different types of T cells, and they have different jobs. And so we're going to have to look at them individually. Remember, B cells, they're going to make antibodies, and remember, this is all about extracellular stuff. T cells is a direct cell-to-cell contact. They're going to destroy a cell, and so it's going to be destroying pathogens that are hiding inside cells. Well, again, there's only one type of B cell, but there are 4 major types of T cells. Well, we need some way to differentiate T cells. We talked about this a little bit last time, but on the surface of these cells are proteins that stick out. And sometimes the exact same protein is always there, and so we can use it like a signpost. These proteins have been named CDs. CD stands for cluster of differentiation. In other words, it's a way to differentiate cells. Now, there are a lot of these CDs, but we're not going to be interested in hardly any of them. A CD, you'll see the word CD, and then there's going to be some number after it. And there's 20, 25 of these different proteins. There's a bunch of them. The only ones that we're going to be interested in, though, are CD4 and CD8, because these are the ones that are found on T cells. And so we can immediately separate T cells based on CD4 or CD8. So here's our normal plain old cell before anything's happened to it. But remember, it's going to grow a surface receptor inside the thymus. Not only is it going to grow a surface receptor, it's going to develop these CDs. And so we're either going to have CD4 or CD8. And so if it's a CD4, they're helper cells, primarily anyway, helper cells. If it's a CD8, primarily they're T killer cells, T cytotoxic cells. There's also another CD8 cell that we're going to look at, which are called T suppressor cells. And then, like B cells, we're going to have memory T cells. So really there are 4 different populations, but these are the 2 that we're going to spend most of our time talking about right here. So again, T helper cells and T cytotoxic or T killer cells. So let's look at these. Well, just like with B cells, we have to go through an activation process. And so when you look at the activation process, we have to remember one thing about it. The receptor is different than it is in a B cell. So a B cell has a receptor that looks like this. And a B cell can interact with an antigen directly. That's what happens when we get sensitization. It interacts with it directly. But T cells can't do that. T cells have a 2-part receptor or a 2-stage receptor, and you have to have what's called co-stimulation. And so T cells don't interact with antigens directly. They interact with antigen presenting cells. And antigen presenting cells are going to eat the antigen, remember, and they're going to put it out here on the surface alongside its MHC. And that is what's going to hook up here, and that's where we're going to get activation. So if you look at a T cell, it simultaneously, at the exact same time, it simultaneously recognizes the MHC, which is self, and the antigen, which is non-self, and both signals are required. It's called co-stimulation. It has to happen. So if you have just one or the other, it won't happen. So if I have a antigen presenting cell that has its MHC on it but no antigen next to it, nothing happens because there's no non-self there. And if I have an antigen just floating around, nothing happens because it has to be attached to an MHC. So when you look at it, it looks like this. Here's our antigen presenting cell. There's the MHC right there. And alongside its MHC, there's the antigen. And in this way, this T cell is presented with both at the same time. So remember, you got to have both. It's co-stimulation. So here's our T cells. Remember, they're going to start out in the bone marrow, but they're not going to stay in the bone marrow. Remember, they're going to go to the thymus, and they're going to mature, and they're going to develop their surface receptor. They also develop this other protein that allows us to separate them. So we have CD4 and CD8, and then they're going to leave the thymus. And then they're going to go through this process of activation, and then we'll see what happens. So let's look at it a little bit more. So remember, they cannot interact directly with antigens. They can't. They're unable to do that. The antigens first have to be processed by an antigen presenting cell, and it has to be attached to its MHC and then presented to the T cell. Well, look, if we look at antigen presenting cells, there are several different types, but one of them we've already talked about, and that's B cells. So B cells have 2 roles, sort of. One is they're an antigen presenting cell, but remember, they're also going to turn into plasma cells and make antibodies. And we already know exactly how that happens. Remember what happens? The antigen is here, it's going to be complexed, and if it's a B cell, it's look like, we'll come back in a second, it's going to look like this. So look, there's our B cell. So we already know it ate the antigen, it brought the antigen inside, processed, and look, there's our antigen right there. And then it went and found a T cell. Remember, we know this is a T helper cell, and the T helper cell released these cytokines and activated the B cell. And then it started dividing and all that. So we already know B cells are a type of antigen presenting cell, but they're not the only type. In fact, they're not even the most common type of antigen presenting cell. Some other antigen presenting cells are macrophages. Remember when we talked about blood, we talked about monocytes. Monocytes, remember, they're the largest of the white blood cells. They tend to have this bilobe nucleus. Well, they turn into macrophages. And macrophages eat stuff like crazy. Well, some of what they eat gets attached to the surface alongside the MHC. And so they're also antigen presenting cells. We looked at dendritic cells just a little bit. They look just like macrophages, except they have lots of little spiny arms. But other than that, they're really just the same kind of cell. They're going to do the same thing. Here's what they're going to do. Here's our antigen presenting cell. And I don't know which kind it is. It could be a macrophage, could be a B cell, could be a dendritic cell. They all work basically the same way. But there's the antigen right there. Remember, this is a free antigen. It's floating around. And so they're going to ingest that free antigen. They're going to combine it with the lysosome. The enzymes in here are going to chop this up into little pieces. Some of those pieces are going to be hung on to, and they're going to be processed and then attached to an MHC. Well, this one is an MHC class 2. An MHC class 2 can only hook up with CD4. So I'm going to process it. I'm going to attach it. It doesn't have to be, though. Let's pretend this is a MHC class 1, because remember, antigen presenting cells have both. So I could also process it with this MHC class 1. Remember, MHC class 1 hooks up with CD8. So I could have 2 on the surface. So there's the one with MHC class 2, and there's the antigen, and then over here somewhere is the one with MHC class 1, and there's the antigen. And what's going to happen is we're going to present the antigen to T cells. Well, CD4, that's a T helper cell. CD8, that's a T cytotoxic cell. But in this case, it's a CD4 that's going to have to do it, because this is MHC class 2. And this is what it looks like. So there it is, MHC class 2. There's the antigen. So this T cell, which has to see both of them at the same time, remember, it's co-stimulation, can hook up right here. And what happens when they hook up is we're going to get activation. Again, here's the same thing. So somewhere over here, this one ate an antigen. It came in, it got processed, it got attached to the MHC. Look, there's 2 of these, though. This is MHC class 2, but that's MHC class 1. But this one, MHC class 2. There's the antigen right there. And then here comes the T cell. T cell then can interact with the MHC and the antigen. That's exactly what's going on here. Remember, it could have been a B cell. So that B cell ate the antigen, processed it, put it alongside its MHC class 2, If it's MHC class 2, what's the only cell that can hook up with? What CD matches MHC class 2? Anybody know? CD what? 4. Which cells are CD4? T helper cells, CD4. So that's going to happen. We're going to release the cytokines. This is now activated. It's going to go through all that process that we've talked about before. Same thing here. So here's a little sort of summary cell of it. There's our antigen presenting cell. This one happens to be probably a macrophage, but it definitely is a phagocyte. It's going to eat it. It's going to process it. It's going to put it on the surface, and then it's going to go find a T cell. And once it finds the T cell is activated. Well, when you activate a T helper cell, T helper cells do more than one thing. They activate B cells, but they also help activate T cells, T cytotoxic cells. When you activate T cytotoxic cells, though, they go out and they destroy infected body cells. And then when a T suppressor cell is activated, T suppressor cells are going to tamp down the immune system. So look, 3 very different roles here. One is a helping role. One is sort of a killer kind of role, killing role. And then this one is sort of a suppressing role. In other words, it brings everything back down to normal. But let's look at T helper cells first. So T helper cells, remember they're regulatory cells. And then remember they regulate this response by these cytokines. And so they release the cytokines and they can activate other cells. They do it by activating these other cells by chemicals. And so they sort of play a central role. Because if you don't have any T helper cells, we don't have any of these cytokines. If you don't have these cytokines, there's no activation of these other cells. And if you don't have an activation of cells, you get no immune response. So if we go back to our analogy of the military, T helper cells are sort of like the generals. In other words, they tell everyone what to do. They tell you to go over there and do this, and so on. If you take out all the generals of the military, you probably would not have a very effective military. Its effectiveness would drop like crazy. And if you were to take out all of the T helper cells, that's exactly what would happen. There would be no immune response. And so, no immune response, this person's going to get sick very, very easily from just about anything. But how do we activate a T helper cell? It looks something like this. We're going to hook up with an antigen. We have to have this antigen presenting cell that comes along. In this case, it's a B cell. And then we're going to get this co-stimulation. Not only are we going to activate this cell, but we're also going to activate this cell. And so not only is the B cell going to divide and make plasma cells and memory cells, but we're also going to have the T cells divide. Let's look at T cytotoxic or T killer cells. So remember, these directly attack and kill other cells. And here's what their targets are. Their targets are cells that are either foreign are cancerous or they're hiding something on the inside. Well, they also have to be activated. So here's our antigen presenting cell. And look at it. Remember, antigen presenting cells have both MHC class 1 and MHC class 2. So look what they can do. They can hook up with this. There's a CD4. But they can also hook up with this. That's a CD8. So they play the central role in bringing these two close to each other. And what happens is then we're going to release those cytokines and we're going to activate this cell. So they now become activated. So think about this. Can a T helper cell hook up directly with a T cytotoxic cell? Remember, this one has CD4 on it. And CD4 can only hook up with MHC class 2, but this one has CD8 on it. And it can only hook up with MHC class 1. It would be very awkward for these two to hook up. And so that's where we have the antigen presenting cell playing this sort of central role. It's a little bit like a matchmaker. It allows these two cells to come into close proximity so this helper cell can activate this T cytotoxic cell. Once you activate a T cytotoxic cell, just like when you activate a T helper cell, we're going to go through mitosis. So remember what happened with the B cell? It immediately started dividing, and we went from 1 to 2 to 4 to 8 to 16 like that. That's exactly what's going to happen here. And that's exactly what's going to happen here. We're going to make a bunch of clones. We're going to make a bunch of clones. And so very quickly, we're going to go from an army of just 1, maybe 2, to an army of a huge number. So we're going to have a whole bunch of T helper cells, and we're going to have a whole bunch of T cytotoxic cells. The T helper cells go out, and they help B cells, and they help other things. They help T cells to activate. Well, T cytotoxic cells are going to go out and kill. So there's the T helper cell. There's those clones. And then T cytotoxic cells, when you look at them, remember this word means cell poison is what it means. They're killer cells. And they're going to go out and kill these entire cells. So they're activated. But the question is, how do they know which cells have these antigens inside? You can't see in there. How do you know? So something has to be able to do, or they have to be able to do something to be able to know whether cells have bacteria or viruses or something on the inside, because they're going to go out and they're going to go out and kill. But you don't want to kill the wrong cells. You only want to kill the ones hiding the antigen. And so there has to be some way for them to know which cells are hiding the antigen. So look at this. This is a body cell. This is not an antigen presenting cell. It's just a plain old body cell. Could be a skin cell, could be a muscle cell, could be a fat cell, could be epithelial cell. It's just a plain old cell. But this cell is unlucky because look what's happened. Something has gotten inside and it's going to take over this cell. It's either going to eat it sort of from the inside out, or it's going to turn it into some sort of virus factory or something like that, but it's going to take over. But before that happens, this plain old normal body cell is going to do something sort of sneaky. As this thing grows in here and grows, some little pieces are going to be processed and they're going to be put on the surface. So look what's going to happen. Some of these little pieces are going to be processed and they're going to be attached to our MHC, and then they're going to be put on the surface. Well, this has to be MHC class 1, because remember, the only cells that have MHC class 2 are antigen presenting cells. This is not an antigen presenting cell. This is a body cell. Remember, all nucleated cells have MHC class 1. But look what happened. This cell put this on its surface. It's sort of like painting a big red X on your door. In other words, that big red X says that there's some bad guy, something hiding behind this door. And what a T cell is going to do is it's going to come along and find that. So look, we have a whole bunch of cells here. But only this one cell was infected. Only this one cell got in there, the antigen. So the antigen grew. So what did this cell do? It does this. So look, it puts this big red X on the door. Well, when T cells come along, there's nothing there. Nothing there. But when they get here, look, there's that big red X. And so this T cell is going to then kill that particular body cell. So look, there's activation. We know about that. And then the T cell goes out. Remember, we're going to have hundreds of these. They're all going to look for the exact same antigen. Here's this body cell. Look, it's infected by a virus. So what it did is the virus that's in here, remember, it's going to take little pieces of it, process it alongside its MHC, and put it on its surface. And look, again, it's like painting a big red X on the door. That antigen is the one this T cell was activated for, and so it fits. And then remember, this is a CD8, and this is MHC class 1. They fit. And what's going to happen then, that T cell is going to destroy this infected cell. It's going to kill it. Does it make sense? Anyone have any questions? Well, let's see how it kills it. So there's our infected cell. There's our pathogen, antigen. It got in. Remember, we're going to be able to process, but before this cell is completely destroyed or completely taken over, takes a little bit of it, puts it on its surface. Again, that's like painting a big red X on your door. Here comes the T cell. It's a T cytotoxic cell. It's CD8, which matches MHC class 1. When they see that antigen there, then this T cell is going to release poisons, toxins. And those toxins are going to kill this cell. Some of the toxins cause lysis. In other words, a cell is going to pop or rupture. Some of them are enzymes which chew up the inside of the cell. And some of them are just literally poisons. So it looks like this. There's the T cell, look, there's a virus-infected T cell or a bacterium-infected T cell or a cancer cell. Well, if it matches with one of these, it's going to kill this cell. And it looks like this. So there's our antigen presenting cell. Remember, it's going to go out. It's going to find, here's a T cell. There's a helper cell. There's our T cytotoxic cell. We're going to activate this T cytotoxic cell. We're going to get a bunch of these. They're going to go out looking for that antigen, that pathogen. And that thing infected this cell right here. But again, before it completely was killed, look what it did. It put pieces of that pathogen on its surface next to its MHC. And here comes this T cell, finds it, releases these poisons and toxins, and kills the cell. Remember, some of these are lysins. In other words, they're going to produce holes in the cell, and they're going to cause the cell to rupture. Well, this is very similar to what we saw with complement, but it's a different protein. But it creates holes, pores. And what happens when you have all these pores is stuff goes in and stuff goes out. The cell dies. Any questions? So there comes our T cell. There's our infected body cell. They're going to come right up next to it. They actually touch. Remember, this is actually called the kiss of death. And so when they touch, when they kiss, we're going to release these poisons, these toxins. Some of these are going to create these channels. Stuff is going to go in, stuff's going to go out, and then we're going to destroy the cell. And here's actually happening. So look, at some point, we're going to get rid of the antigen. So remember B cells? They're going to go through that whole process. We're going to make a ton of antibodies. The antibodies are going to float around. And the antibodies are going to get rid of free antigen. In other words, all the antigen that's floating around in your blood or your body fluids, these antibodies are going to get rid of. T cells, on the other hand, remember, they can't interact with free antigen. They can only interact with the cells that are expressing the antigen next to their MHC. But remember, any cell that is expressing that, that means that it's an infected cell. And so T cells are going to destroy those. So they're going to get rid of the antigens inside cells. And so slowly, all of the antigen is going to be removed. So antigen levels are going to drop. Well, think about this. If you have no more antigen, does it make any sense to keep making more antibodies or keep making more of these T cytotoxic cells? It's like if you no longer have a war, it's like you continue to build up your military. It makes no sense at all. What usually happens is when a war is finished, we decrease the military. And that's exactly what happens here. But it takes another whole kind of cell to make this happen. And that is a T suppressor cell. So T suppressor cells are also regulatory cells. They also make cytokines, just like a helper cell does. But they do the opposite thing of a helper cell. They don't activate, they inactivate? Well, as the antigen goes down, T suppressor cell number goes up. And what T suppressors are going to do is they're going to basically tell everybody to calm down. There's no reason to keep going. The battle is won. It's all over with. And so they're going to release chemicals that inhibit T cells and B cells. In other words, they basically tell them their job is over, it's time to rest. But again, it's by these cytokines, which are basically just little local hormones. Any questions about that? Well, that was a very brief discussion of T cells. But again, T cells are a lot more complicated than B cells are. And the one reason is because they have 4 types. Another reason is that you have to have this co-stimulation. They can't directly interact with antigens. Another thing is that they're going to destroy cells. So if you ever take an immunology course where you spend a whole semester, you'll spend a whole lot more time talking about T cells. Even if you take micro, you'll probably spend a whole lot more time talking about T cells. Any questions though? Any questions about T cells or B cells, the different types of T cells, how they work, antigen presenting cells, what they do, how they work? Any questions about any of that? Okay, well, obviously we want the immune system to work. We want it to get rid of antigens, but there's things that can go wrong with the immune system. And one of the things that can go wrong with the immune system is when it's just a little bit too active. In other words, you develop what's called a hypersensitivity. So the immune system sort of goes crazy. And usually what it goes crazy against is something that is completely harmless. Look, this is pollen. And pollen does absolutely nothing to the human body. It's really just a plant sperm is what it is. It doesn't hurt you in any way. We shouldn't react to it. Our body should just ignore it. But that doesn't always happen. And so when you get this hypersensitivity to these normally harmless substances, we call it an allergy. So let's look at allergies just a little bit. There are actually 4 different types of allergies, and they're just designated type 1, 2, 3, and 4. Well, you can tell them apart because it might be antibodies involved or it might be T cells involved. Also, it might happen very, very quickly or it might be delayed. You can also tell by what kind of antibody is involved. So is it IgE? Is it IgM? Is it IgG? And so we can differentiate or separate them out. So let's look at type 1 first. So type 1 is called anaphylaxis, but there's actually 2 types of anaphylaxis. One type is local anaphylaxis. And so local means that whatever happens is just in a general area of the body. So it's just in your arm, or it's just in the leg, or it's just in the intestines, or whatever. But it's not life-threatening. It's not life-threatening. It's just irritating. So what you get are a bunch of symptoms that are very, very unpleasant, like itchiness, runny nose, red eyes, watery eyes, maybe hives, something like that, something very, very local to the area. And it depends a lot on how the allergen gets into your body. If it comes in by inhaled stuff, you're going to have respiratory symptoms. If it gets on your skin, you're going to have skin rash. If it gets on in your intestines, you're going to have something like diarrhea or cramping or vomiting. Again, all of these are unpleasant, but they don't kill you. And they're almost completely caused by histamine. And so histamine, at least in local areas, what it does is it causes a local inflammation. And remember, there's all these cardinal signs of inflammation: heat, redness, swelling, and pain. You're going to get those things. So if we can get rid of the histamine, or if we can counteract the histamine, we can stop this from happening. And so we have a whole class of drugs called antihistamines, which basically block the effects of histamine. All they do, they block the effects of histamine. And so I don't know how many of you have like hay fever or cedar fever or anything like that, but you know how you feel when you get those symptoms. You just feel lousy, but it doesn't kill you. I'm allergic to cats. So cats, they have a protein in their saliva that I'm allergic to, and they lick themselves like crazy. And then what happens is they shed skin particles, and the skin particles, the dander gets into the air and on the furniture and places like that. Well, when I get near that, I have a very bad local anaphylaxis. My eyes get swell up and they get itchy and watery. My nose runs, but I don't die. I might wish I'm going to die, but I don't die. And if I know that I'm going to be around a cat, if I take an antihistamine ahead of time, I can pretty much get rid of these symptoms. So that's a type 1 local. And so a lot of people are allergic to multiple things, and so they go to a doctor and they have a skin allergy test. And so basically all this is they inject you with the allergen. Now they also inject you with histamine. Histamine is what's called a negative control. In other words, that's what histamine does. So look, that caused a histamine release. That caused a histamine release. That may have caused a tiny bit, but not much. And so what they do is they compare each one of these to the control. But there's another kind of anaphylaxis, and this is called anaphylactic shock or systemic anaphylaxis. And systemic anaphylaxis or anaphylactic shock can kill you. It can kill you. It works exactly the same way as systemic, but the problem is it happens all over your body at the exact same time. It usually happens because the antigen somehow gets directly into your blood. And so it travels all over the place, and which means all over the place, we're going to get histamine released. And so histamine has all those effects, but since it's system-wide, it's multiplied in terms of how bad it is tremendously. And so if it's in when that histamine is in the lungs, you can get things like constriction of bronchioles. If it's in blood vessels, you can get vasodilation. Fluid leaks out, you get swelling like crazy. And then depending on where the swelling is, it can make your throat swell up or your tongue swell up. And so you could have trouble breathing, or you could have trouble with low blood pressure because the vasodilation happens all over the place. And those things, especially blood pressure, and of course breathing, if you can't get air and you can't have blood pressure, you can't live. And so you go into what's called hypotensive shock, and then you could die from it. Well, you don't have time for an antihistamine to work. It just doesn't work quickly enough. And so if you are going into shock, you need something that works really, really, really fast. And so the treatment of choice is epi. So a lot of people carry EpiPens. And so all that epi does is counteract these effects. So epi causes the bronchioles to dilate. Epi causes your blood vessels to constrict. You're going to get vasoconstriction. You're going to increase your heart rate. So you're going to increase your ability to breathe, and you're going to bring your blood pressure back up. It also has a bunch of nasty side effects too. If you've ever taken it, you would know. But this is how this is what the bronchioles look like. Look, those are normal. But look, that's what happens. You get constriction. You also get a huge release of mucus. And so it completely blocks these bronchioles. Look how much air could get through here versus how much air can get through there. And then blood vessels dilate like crazy. Capillaries dilate as well, and they become leaky. And so fluid leaks out like crazy. And so you get swelling. And then when all the blood vessels dilate, remember, we don't have enough blood to fill up our blood vessels, and so blood pressure drops. Well, this is how it works, and it's similar to what we've seen before. Look, there's an antigen. The antigen gets into your body, and we're going to process it just like we always have. We're going to do the thing that we talked about, and so we're going to get B cells. And so we're going to get a ton of B cells. And then the B cells are going to turn into plasma cells, just like we've talked about before. But here's the difference. The antibody they make is IgE. And remember we talked about IgE when we talked about the different types of antibodies? It's the only one that can attach itself to basophils and mast cells. So remember, basophils and mast cells have tons of granules, and those granules contain histamine. So what happens the next time you encounter this antigen, this histamine is going to be released. So it looks like this. There's the second time, the next time, the third time, the fifth time, or whatever. It's going to attach here, and we're going to release histamine. Well, these cells are not in one place. They're everywhere. And if they all release histamine at the same time, what's going to happen is all this histamine is going to cause those effects. So we're going to get a lot more mucus produced. We're going to get bronchial constriction. We're going to get vasodilation. Fluid's going to leak out. We're going to go drop in blood pressure. And then this is going to make you unable to breathe. So if you don't do something pretty quickly, you could die. Make sense? Any questions? So that's type 1. Let's look at type 2. We've also already talked about type 2 because type 2 is what happens when you get a blood transfusion of the wrong blood type. Remember we talked about blood types. We said we had type A, and we had type B, we had type AB, and we had type O. And we went through who could give to whom. And remember what happens if you give the wrong blood type to the wrong person. If I give type A to someone who's type B, what's going to happen in that type B person is we're going to get that clumping. And the reason we get clumping is because of those IgM antibodies. So remember, IgM is a pentamer. And so when you have a pentamer, you got 10 binding sites. 10 binding sites, which means that you can connect things together really, really, really easily. And so we connect red blood cell with red blood cell with red blood cell with red blood cell. This is not a clot, remember. It's a clump. It's a agglutination. So type 2 is an agglutination reaction. Let's look at, and here's those blood types. And look, there you can see what's happening in there. So you can see the clumps. But the clumps in here are tiny, but they don't have to be tiny. They can be huge. So if you look inside someone's blood vessels, they clump up like that. But these others are also going to clump up. And we're going to get clumps that are like this. Even bigger clumps and bigger clumps. And they're going to block blood vessels. They're not going to flow. And so you're going to wind up also with death. Let's look at a type 3. Type 3 is called immune complex hypersensitivity. So remember when we talked about the ways that antibodies can get rid of antigens, one of the ways that we said was that they can surround it like this and neutralize it. But another way that they can do it is they can form chains. So you get a chain. And so what you wind up with is something too heavy to be soluble anymore. And so it precipitates. And so what happens is these complexes fall out of solution. And wherever they fall out of solution, here they are right here, white blood cells are going to come to clear that area. But when the white blood cell comes to eat this, most of the time, not only do they eat that, but they also damage this tissue that it's sitting next to. And so you get this sort of peripheral or collateral damage to healthy tissue. And depending on what organ they're in, that organ is going to be damaged badly. Could be your skin like this. But this can also be on the inside. So if you think about inside your gut, what if those immune complexes form here? Well, we're going to destroy the gut wall. It's in your kidney. You can destroy the kidney. And so one of the main conditions that comes from this is lupus. And so if you think about lupus, lupus can destroy any tissue because it just depends on where these complexes wind up. And then what happens is you get this very, very intense inflammation of that tissue. And so cells die. So Look, there it is in the gut, and here are these complexes right here. So white blood cells are going to come, but not only will they destroy that, they'll destroy this as well. All right, we have one more type, and this is called delayed type hypersensitivity, or it's also called type 4. It's delayed. It takes some time for it to happen. And so it takes somewhere between 24 to 72 hours before you start seeing any kind of problem. Well, this is not antibodies. This is T cells, T cytotoxic cells. What's going to happen is the T cytotoxic cell is going to destroy cells just like it always does. And so remember, if you had something hiding in here, you would destroy that cell. But you could also destroy a cell that belonged to somebody else. They have the wrong marker on here. So if you get something like a transplant of someone else's tissue, your own T cells might destroy that transplant. Another example of this, and more common example, is what happens if you get poison ivy or poison oak. Poison ivy and poison oak and poison sumac have an oil in them. It's called urushiol. And that oil, that urushiol, at least for most people, their bodies hate it. And we hate it so much that if this gets a tiny little drop on a cell, T cytotoxic cell is going to come along and kill that cell. So any cell that touches or is touched by this urushiol will be destroyed. So it could be on your skin. Most people, that's how they come into contact with poison ivy. And so wherever they're going to come into contact with that oil, we're going to come and destroy it. So you're going to get a very, very nasty rash. Well, histamine is not the problem, so antihistamines don't work. The only thing that does work is some sort of steroid, like corticosteroid, like cortisol or hydrocortisol or something like that. But think about this. What if it's not on your skin? What if you got it in your mouth? So you got it in your mouth. Somehow you ate it. So it's going to destroy all the tissue in your mouth. What if it went down your throat? You swallowed it. It's going to destroy all the tissue as it goes all the way down. So you would have this horrible sort of rash on the inside. What if you breathed it in and it got down into your lungs? Well, guess what? The same thing's going to happen to your lungs. Not everybody is allergic to poison ivy, though. About 75% of people are, but about 25% of people are not. Remember, it can be poison ivy, but it can also be tissue rejection from a graft. So you probably heard about the face transplant. Well, that's basically what happened with this guy here. But his body rejected it. Same thing happened here. This was a burn graft. So the burn graft from someone else got rejected. There's a kidney rejection from a transplant. And then, of course, you can also get it on the skin. Any questions about the different types of allergies? Okay, well, that's if you have a hypersensitized immune system. But you can also have the opposite thing. You can have a deficiency in the immune system. You can also have the immune system attack your own body cells. So you can have what's called an autoimmune disease. And we looked at some autoimmune diseases already when we talked about hyperthyroidism. One of the most common types of hyperthyroidism is an autoimmune disease called Graves' disease, but also things like rheumatoid arthritis and multiple sclerosis. So what happens here? What happens is we forget the difference between self and non-self. In other words, remember those cells, they were tested. They went through positive and negative selection, but somehow they escaped. Somehow they either cheated on the test or didn't take the test at all. They escaped. And so what is now wandering around in this person's body are all these cells that don't know the difference between self and non-self. And so they attack the body's own cells. We can also have where the cells don't work. Remember B cells, they mature in the bone marrow. T cells mature in the thymus. But some people are born without a thymus. They don't have one, which means they have no T cells, no functioning T cells. Some people have bone marrow issues and they don't have B cells. Some people are born without either one. They have no B cells and they have no T cells. I don't know if you've ever heard about the boy in the bubble, but that's what he was. He had no B cells and no T cells, which means basically anything could kill him. Well, they figured this out right after birth, and they basically put him in a sterile environment. His room was a sterile environment. He could never touch another person. He could never, then he lived basically inside this box, basically. And in order to touch him, you had to use these gloved hands that went through these portals. His food was sterilized before it was given to him. There wasn't a single thing in it that was alive. And he could leave his house, but he had to leave his house in kind of a hamster ball. And so he spent his whole life basically not touching anybody. He was isolated. Well, when he got to a certain age, in his 20s, he decided he didn't want to live that way anymore. And so he left his bubble. What do you think happened to him? He died, exactly. And it didn't take very long. They made a movie about it. You can watch it. It starred John Travolta. But it was, again, about the boy in the bubble. What he had was this. It's called severe combined immunodeficiency. He was born with that. This is another one you're born with. But you can also acquire an immune deficiency. An acquired immune deficiency, it means you're not born with it. You get it later in life. And that's where this comes into play. This is caused by a virus. And the virus is HIV. And so this generation knows a lot about HIV, but my generation we were young when HIV came along. It was in the 80s. And so there were tons of people who died from this before there were any treatments. In fact, we lost a whole bunch of people, basically a lost generation of people from this virus. But the virus, what makes it so deadly is what it infects. Remember we talked about this whole Army and Air Force thing? Well, we went through the whole cells. There were B cells, cells, and then there were T cytotoxic cells, and they were T helper cells. Also, we had T suppressor cells. But think about this. There's one of these cells that basically helps or is involved in activating these others, and it's this one. And that is the cell that primarily HIV infects. So if you get rid of all the T helper cells, B cells, no matter how many of them you have, they don't work. And T cytotoxic, no matter how many of them you have, don't work. They don't do anything. And if they don't do anything, they can't protect you. And so what happens once you get rid of all these T helper cells is whoever has this disease gets multiple secondary infections. And that's what they die from, these secondary infections. But if you look at this virus, it infects these T cells, and again, the T cells then are slowly destroyed. Any questions? And then if you look at someone who has these no T cells, they don't die from lack of T cells. What they die from is some other infection. So it could be a cancer, could be some sort of pneumonia, could be encephalitis, could be this wasting disease. Any questions? So, that's the immune system.